Hello, ladies and gentlemen. I would like to welcome you to lesson 5. Our topic is chemicals of life, and in this particular lesson, we are going to look at polysaccharides. The word polysaccharides simply means poly means many, and saccharide means sugar units. So simply polysaccharide means it's made up of many sugar units, or it's made up of many monomers combining together to form a large complex molecule called the polysaccharide. So I want us to understand a little bit more about them. So many monosaccharides may combine through a type of reaction called condensation reaction to form polysaccharides. Remember, we looked at condensation reactions when we looked at the formation of the disaccharides. This is a reaction where two small molecules combine and water is produced, is lost through this combination. That kind of reaction is called condensation reaction. A combination of two or more smaller molecules with the loss of a water molecule is what we call a condensation reaction. So similarly, in the formation of the polysaccharides, condensation reaction is the most preferred form of reaction. A number of monosaccharides which combine may be variable in number. So the number of monosaccharides that combine to form a polysaccharides may vary in number. They may also be branched. In some polysaccharides, the monomers are branched, or in some cases they are not branched, they are unbranched. Yes, we shall see a little bit later on. But for now, let's look at the properties of polysaccharides. Remember, we looked at properties of monosaccharides. You remember them? We also looked at properties of disaccharides. So today we want to look at the properties of polysaccharides. The first one is that they are not sweet. I hope you have tasted cassava of recent. You have eaten maize. So those are some of the components that contain polysaccharide starch. Yeah, so they may have their specialized taste, they may have their characteristic taste, but they may not be sweet. For example, compared to sugar, which is a disaccharide, you realize that the polysaccharides are not as sweet. And then they also do not dissolve in water, so they are insoluble in water. They cannot be crystallized. So if you grind them, if you pound them, you realize that they form powdery substances. They don't form crystals. And then they have, of course, a very high molecular mass. Being a polysaccharide, it means it has very many monosaccharides. So therefore, the molecular mass is increased depending on the number of monosaccharides involved in the formation of this polysaccharide. And then they are non-reducing sugars. So if you react them with Benedict solution, they do not change the color of Benedict solution to orange or red or brown. So that means they are non-reducing sugars as well. And then the chains may be folded to make them compact, which are ideal for storage. Such a large size of molecules make them insoluble in water and suitable for storage, as they exert no osmotic influence and not easily diffuse out of the cell. So of course, being large molecules, they do not really have impact on osmotic movements of fluids in cells. So therefore, they are a very good storage compounds, as we shall see later on. And an example of a good storage polysaccharide is starch, which is found in plants. So starch is the main storage material in green plants, while glycogen is for animals. Of course, we had mentioned this earlier, that the main storage polysaccharide in plants is starch. And then for the case of animals, it is called glycogen, and usually highly concentrated, especially in the liver, which is the metabolic center, but also in the muscles, where metabolism takes place. Upon hydrolysis, when these polysaccharides are broken down, they are broken down into their constituent monosaccharides. For example, if starch is a polysaccharide of glucose, as it is actually, starch is a polysaccharide of glucose, alpha glucose. So when you break down starch, you produce alpha glucose molecules. So therefore, the process by which a large polysaccharide or a polysaccharide is broken down to produce its constituent monosaccharides is what we call hydrolysis or breakdown reaction. A reaction of breaking down a larger molecule in the presence of water is what we call hydrolysis. Of course, not all polysaccharides are used for storage. E.g., cellulose is a structural polysaccharide giving support and strength to the cell walls. So some polysaccharides are used for storage, others are support structures, as we shall see later on. So let's start with the starch. An example of polysaccharide. We are going to look at majorly the common polysaccharides that have biological significance in living systems. We may not exhaust all the polysaccharides, but we shall look at the common ones that we ought to know. One of them, of course, is starch. And like we mentioned earlier, starch is found in plant parts in the form of starch granules. So it is usually stored in the plant cells as starch what? Granules. And it's a reserve food formed by any excess glucose during photosynthesis. So after photosynthesis has taken place, the first product there is glucose. But in case glucose is not used by the cell at that moment and is excess, it can easily be converted to starch and stored as starch granules in plant cells. It is usually common in seeds, for example, in maize. You have seen that in millet, in most of the cereal seeds, and then also in some roots like the tubers, like potatoes or cassava, yams. You can easily find starch there. So let's look at a little bit more about the structure of starch for now. As we mentioned earlier, of course, starch is a polymer, and is a polymer of alpha glucose molecules. And these glucose molecules in starch are held together by what we call glycosidic bonds, forming the chains of alpha glucose units, which get folded or coiled into a helical structure called a helix, a highly folded structure. So what we need to know is that starch is made up of very many alpha glucose molecules combined together through condensation reactions. The bonds between these glucose molecules is called the glycosidic what? Bond. And we had mentioned this earlier in the formation of disaccharides. And we have also said that this starch molecules, this monomers, these alpha glucose molecules, in case there are so many, they can be coiled. They coil and they form what we call a helix, a helical structure. The other most important point for us to note is that starch has two components. The first one is called amylose, and the other one is amylopectin. Yeah. 20% of starch is usually made up of amylose, and 79% is made up of amylopectin. And then other substances can be there. In case they are there, they form up to about 1%. For example, the phosphates and the fatty acids. So, in the structure of starch, we have two polymers. We have amylose, which is about 20%, and then amylopectin. Amylose, in case you want to test it chemically, it stains deep blue with iodine. In case you drop iodine into a solution of amylose, you will see a deep blue color, meaning amylose is present in that solution. Then for the case of amylopectin, when you drop starch in a solution of amylopectin, it will stain red or reddish purple. That is how we can differentiate the two. In case you have amylose and amylopectin as separate solutions, when you drop iodine solution in amylose, you will form a deep blue solution, and if you dropped it separately in amylopectin, it forms a purple solution, or purple to red, a blend of purple and red. Yeah. Amylose is structurally unbranched, while amylopectin is highly branched. That is one of the key differences between the two polymers of starch. Amylose is a straight chain, it's not branched, while amylopectin is highly branched. So you can see there the structure of amylose, and you see we say it is unbranched. It is a continuous straight structure. Of course, between each glucose molecules, there is a formation of alpha 1,4 glycosidic bonds, the bonds that form between two alpha glucose molecules in that amylose structure. So you can see that straight chain there, part of the amylose structure there. And then for the case of amylopectin, you see a much more complex structure. The difference between the two is that amylose is highly branched, as you can see, and also has different types of bonds. It has alpha 1,4 glycosidic bonds, but also has alpha 1,6 glycosidic bond, the bond that forms between alpha glucose carbon atom 1 and carbon atom 6 of another alpha glucose. That is 1,6 glycosidic bond. Then for the case of 1,4, a bond between carbon 1 of one alpha glucose and carbon 4 of the adjacent glucose molecule there. So you can see the differences between amylose there and amylopectin there. All right, let's proceed a little bit. That is starch we are discussing, members. So if we are asked that how does the structure of starch relate to its functions, what would you say? Of course, we had said earlier on that the structure of starch, sorry, the function of starch is majorly for storage. That's what we had said. So if starch is used for storage, how does the structure of starch enable the functioning, enable it to perform that function of storage much more effectively? So we can have a summary of our answers here. The first one is that it's a polymer of alpha glucose molecules, hence a large molecule, making it relatively insoluble in water, hence an ideal storage molecule. If starch was soluble in water, it would not be easy to store it. So because of the long polymer, because of the complex structure, it makes it insoluble and therefore very much a viable candidate for storage. The other one is the alpha glucose molecules in starch are held by the glycosidic bonds, which can be broken down to free glucose molecules from the stored starch for ATP synthesis during respiration. Of course, we know that this starch is not useful if it cannot be retrieved back. It's like if you have a granary at home and after storing your food, then you cannot access it back. Then there is no advantage of storing that food. The granary should be able to be accessed when you need the food. That is literally. So starch similarly relates to that. Starch should be able to be broken down to produce glucose, which is a respiratory substrate for plants and actually for organisms. So therefore, its ability to be broken down makes it a very good storage component there. But of course, it's only broken down in presence of enzymes into glucose to produce, which is used for synthesis of ATP during respiration. The other advantage is that the starch molecule is coiled into a helix with a hydroxide group projecting interiorly, making it insoluble in water, hence exerts no osmotic effects on cells and is ideal for storage. When you look at the structure of starch very well, you realize that the hydroxide groups are internal. And remember, the hydroxide groups are the most reactive groups there in the starch, in the glucose molecule that forms starch. And therefore, when it is projecting interiorly, meaning it cannot easily be accessed by reactants that are on the surface of starch molecule. So therefore, it is insoluble. Therefore, it cannot be reacted it cannot also dissolve in water, and that's an advantage. And because of that, it does not exert any osmotic pressure in cells, and that of course is very vital for storage. The chains of alpha glucose units can be folded to form starch grains for easy storage. So those long chains you see there are all fitted within the starch granules that are formed within the cells. You can also add more. That is just, I am showing you the trend on what the examiner may be interested in. The question was relating the structure of starch to its function. So you can continue studying the structure of starch, and then you suggest the suitability of that structure to perform its functions. Let's go to the next polysaccharide here, which is glycogen. And we know, of course, that glycogen is a major polysaccharide storage material in animals. Starch is in plants. Glycogen is in what? Animals. And is majorly stored in the liver and in muscles. Yes. It is also made up of alpha glucose molecules and exists as granules. They call them glycogen granules, and they are usually found in animal cells. However, its chains are shorter. Yes, about 10 to 20 glucose units, and is also branched, just like amylopectin. So, unlike starch, glycogen molecules are shorter. Its chain is a little bit shorter than that one of starch, and then it is much more soluble in water compared to starch. Yeah, so we can look at it there. You can see the structure of glycogen. You will see that there is a 1,6 glycosidic. There is first of all a 1,4 glycosidic bond between the alpha glucose molecules, and then in the branching there is a 1,6 alpha glycosidic bond, as you can see there, and quite similar to the structure of amylopectin. Yeah, that is glycogen. But for it, like we mentioned earlier, it has shorter chains, a few alpha glucose molecules, and also a little bit soluble in water, of course, because of the fewer bonds that are found in it. So let's proceed to another one called cellulose, and this is another very popular polysaccharide, and we know that it comprises about 50% of the plant cell walls. And in some plants like cotton, it forms about 90% of the plant's cell walls. So it's a very prominent structure or component of cell walls of plants. It is a polymer of about 10,000 beta glucose molecules, forming long and branched chains, which are parallel to each other with cross linkages between them, which gives it stability and good structural material. So, like you can see later on, cellulose is a much more giant structure and much more stronger compared to most of the polysaccharides, as we see, because of possession of very many bonds and also having cross linkages, as we shall see later on. So let's look at the structure now in detail. We said it's a polymer with straight chains of beta glucose units held by glycosidic bonds, with the OH groups, the hydroxide groups, projecting outwards from each chain, forming cross linkages of hydrogen bonds with adjacent chains. The cross-linking binds the chains together, which associate to form microfibrils that are arranged in larger bundles to form macrofibrils, as we shall see shortly here. So when you look at the structure of cellulose here, you do see that it is very highly linked. It has, first of all, straight chains. Their chains are not branched. The beta glucose chains are not branched, but they are linked together. They are cross linkages, as you can see from there. However, thank you. Credit to Miss Julia Sochen, rest in peace. Great teacher of biology. Yeah, let's proceed. So let's take note. Starch lacks some structural properties possessed by cellulose because it lacks the cross linkages. So the cross linkages you saw from cellulose make cellulose very much stronger than starch. Yeah, so starch cannot match the strength of cellulose. The stability of cellulose makes it difficult to digest and therefore not a good source of food to animals, except those which have cellulose producing microorganisms which live in them in a symbiotic way, e.g. the ruminants, for example the cows, the goats and the sheep. For them, they can eat cellulose containing grass and leaves because they have cellulose secreting organisms that are found in their guts that secrete the enzyme cellulase. However, most of the other animals cannot afford to digest cellulose because they don't have the capacity to produce cellulase. Yeah, they don't have the capacity to produce what? Cellulase. So let's look at the uses of cellulose. Let's go through them very fast. Rayon, which is produced from cellulose extracted from wood, is used in the manufacture of tire cords. So you must have observed the tires very well, and you will see that there are some cords running across the tire, making it rigid and strong. That one is called rayon, and rayon is a product of cellulose. Cotton is used in the manufacture of fabrics. That word is fabrics, and then clothes. Of course, we know very well the cotton containing clothes, our uniforms, the bed sheets, the hankies, and so on. The masks these days are made up of cotton. So, and cotton is a product of cellulose. We said over 90% up to 90% of the cell walls of cotton is made up of cellulose. And then cellophane, which is used in packaging, is a product of cellulose as well. Must have also seen the masking tape, the packaging tape, that is cellophane, is also a product of cellulose. So cellulose is very important there. Another very important product of cellulose is paper. It's a product of cellulose. So we have used the papers for very many years, and that of course is a product of what? Cellulose. Then there is celluloid, which is used in photographic films. It's a derivative of cellulose as well. You must have seen such films. Nowadays, as we grew up, we used to have cameras that had such films. Yeah, those cameras, those films are made up. They have an active component called celluloid. Yeah, which is a product of cellulose. So cellulose is therefore very important. So in summary, we can see the different polysaccharides there made from these monosaccharides. So from the monosaccharides we have is alpha glucose and beta glucose. And alpha glucose is used in the formation of starch, as we can see down. Then beta glucose is used in the formation of cellulose. So that's a summary there. And earlier on we had talked about the digestion of cellulose, but let's talk about it in comparison with starch. And we are saying that enzymes that digest starch by hydrolyzing its alpha linkages are unable to hydrolyze the beta linkages of cellulose because of the distinctly different shapes of these two molecules. In fact, few organisms possess enzymes that can digest cellulose. Humans do not, so we cannot digest cellulose. The cellulose in our food passes through the digestive tract and is eliminated with the feces. Along the way, the cellulose abrades the walls of the digestive tract and stimulates the lining of the digestive tract to secrete mucus, which aids in the smooth passage of food through the tract. Thus, although cellulose is not a nutrient for humans, it is an important part of our healthful diet. Most fresh fruits, vegetables, and whole grains are rich in cellulose. So therefore, we should always eat. They call them fibers. On food packages, insoluble fiber refers mainly to cellulose. Some prokaryotes can digest cellulose, breaking it down into glucose monomers. A cow, for example, harbors cellulose digesting prokaryotes in its rumen, the first compartment in its stomach, as we shall see later. The prokaryotes hydrolyze the cellulose of hay and grass and convert it to glucose glucose and other nutrients that nourish the cow. Similarly, termites, which are unable to digest cellulose by themselves, have prokaryotes living in its gut, on their guts, that can make the meal of wood, or that can convert cellulose in the wood into glucose that is usable for these organisms. Some fungi can also digest cellulose, thereby helping recycle chemical nutrients within the Earth's ecosystem. So members, much as many organisms do not have enzymes directly that digest cellulose, they associate with other organisms that can produce such enzymes, as you can see in ruminants, in termites, and other organisms. However, even in humans, where we don't have any benefit, any direct nutrient benefit from cellulose, it also performs a function still by stimulating the digestive system, the walls of the digestive tract, to secrete mucus, which lubricates and enables passage of the feces, and also protection of the digestive system or the digestive tract or alimentary canal from abrasion from other acids and so on. So therefore, it's important for us to eat foods that have cellulose at some point. For example, fruits and whole grains. So we have a question here. How does the structure of cellulose relate to its functions or to its roles? I want you to attempt that question. How does the structure of cellulose relate to its roles? Remember, we looked at the other question of starch. How does the structure of starch relate to its functions or its roles? And we attempted that together. So this one, I want you to try to attempt it as your assignment number 1, and then you send me your findings. You send me your responses in my inbox. Yeah. So let's proceed to other polysaccharides. One of them is chitin, or some of you can pronounce it as chitin. Yeah. So chemically and structurally, chitin resembles cellulose, but it differs in the possession of acetyl group. That group you can see there, instead of one of the OH groups like cellulose. It has a structural function and is a major component of the exoskeleton of insects and crustaceans. It is also found in fungal cell walls. So chitin is a very popular polysaccharide also, especially it forms the exocuticle of the arthropods. The exocuticle of the arthropods, the crustaceans, are also a very important structural component of the fungal cell walls. It is quite similar to cellulose, but the difference is that it has the acetyl group instead of the OH groups. As you can see from the structure there. And then the other one is insulin. Another polysaccharide is insulin, which you know as a hormone, which is involved, of course, in the regulation of sugar in our bodies, and is a polymer of fructose, and is found as a storage carbohydrate in some plants and even in animals. As you can see there, the human insulin, yes, a polysaccharide of fructose. And then we have a group of polysaccharides called the mucopolysaccharides. So this group includes the hyaluronic acid, which forms part of the matrix of the vertebrae connecting tissue. It's also found in the cartilage, in the bones, in the vitreous, the eye, and the synovial fluid, and even other structures. They are called the mucopolysaccharides. And then we have heparin, which we know very well as an anticoagulant, which contains also mucopolysaccharides. That heparin is found in blood, and it prevents blood from clotting when it is in the blood vessels. So therefore keeps the blood, in a fluid form. So that one is called heparin. Imagine if we didn't have heparin in our blood, the blood would be clotting as it flows through the blood vessel. It will be clotting at any destination. But because of the heparin, it keeps the blood in fluid form as long as it's flowing in the blood vessels. So today we have looked at a wide variety of polysaccharides, from starch to cellulose to insulin to chitin and many other polysaccharides. So I want you to make a summary of all that we have looked at today, and then summarize this in your book, summarize the structures of these different polysaccharides, and also do the assignments that I've given you, especially relating the function of cellulose to its structure. Otherwise, thank you so much. Also, continue staying at home until the schools are safer for you to return to. Thank you. Be blessed.